Hello everyone, and a very warm welcome to KognitoKey. I am absolutely thrilled to have you here today. We all know flowers are just incredibly beautiful to look at, right? They add so much wonderful color and scent to our world. But today we're going to do something a little bit different. We are going way beyond the surface to take a hands-on, inside look at the hidden biological secrets of flowers. In this explainer, we're essentially going to perform a virtual flower dissection. So, let's dive right into this fascinating topic. So, ask yourself for a second, what exactly makes a flower? It's super easy to just admire the colorful petals and those alluring scents, but there is literally an entire world of incredible morphology and intricate reproductive structures hidden right there on the inside. You see, flowers aren't just there to look pretty. Those structures are specifically designed to turn into fruit after fertilization. Today, we're going to pull back the curtain and see exactly how they pull that off.
If you want to follow along at home, you really only need a few simple things. Here is your dissection toolkit. Grab a whole flower. Something like a pink lily or a China rose is absolutely perfect for this because their parts are very clearly defined. You'll also need a blank piece of paper, a marker or pencil for labeling, some simple cutting tools like a scalpel and a pair of scissors, and maybe some tape to carefully secure your parts to the paper once we separate them.
Okay. Getting started is the fun part. Once you have your blossom, I know it's tempting, but resist the urge to cut right away. First, just use your sense of touch and sight to closely observe the outside. Check out the outside stalk that's holding the flower up. That is called the peduncle. Right above it, you'll spot a thickened base called the receptacle, or thalamus. This is literally the foundation from which all those delicate floral parts grow. Finally, just prep your paper with some labels so we can organize our findings as we go.
All right. As we look at the very outermost layer, we find our first key structure, the sepals. These make up the outermost world of the flower, which is collectively known as the calyx. Usually, these are smooth leaf-like structures. Their main job? They act like a sturdy little shield, protecting the delicate flower while it's still developing in its budding stage. Once the flower finally blooms, they either peel back or just drop off completely.
Now begins the actual dissection. Take your scissors and very gently snip away those sepals. If you are using a China rose, you're going to notice the sepals are distinctly green. But if you have a pink lily, something kind of amazing happens. The sepals are actually identical in color to the petals. Pretty cool, right? Once those sepals are completely removed, you can go ahead and carefully snip off the bright petals.
And that brings us to, without a doubt, the most famous part of the flower, the petals. This brightly colored secondary whorl forms what botanists call the corolla. Now, the crucial thing to remember here is that these vibrant colors and unique textures aren't just for us to enjoy. They actually act as brilliant, flashing advertisements. They are specifically designed to attract insects and other really important pollinators right to the plant.
So, as we peel back those petals, we finally reach the innermost whorls. These are the true reproductive engines of the flower. First up, we encounter the stamen. This is the male reproductive part of the flower. It consists of two main pieces. You have a long, slender stalk called the filament, and sitting right on top of that is a little knob-like structure called the anther. And it's inside this anther that the all-important pollen is actually produced. Take your scissors and gently snip away the stamens. Once you have one free, go ahead and tap the anther right onto your paper. You will actually see the tiny pollen grains fall out. If you happen to have a magnifying glass handy, take a really close look at the shape of those grains. These are the structures that give rise to the male gametes. After you've done that, go ahead and separate the filament from the anther on your labeled paper.
Now, right in the very center of the bloom, standing tall, is the female reproductive counterpart. This is called the pistil or the gynoecium. It has three very distinct sections. Up at the very top is a sticky, platform-like structure called the stigma, and that's designed specifically to catch and collect the pollen. Right below that is a hollow tube called the style, which carries the pollen downwards. And at the very base is the ovary, where the flower's eggs are safely held, just waiting to be fertilized.
Okay, this next bit is the most delicate part of our whole dissection. Take your scalpel and very gently slice the pistil lengthwise, right down the middle. When you open up that ovary at the base, grab your magnifying glass again. You will be able to see the tiny, hidden ovules inside. It honestly feels a bit like finding hidden treasure. Once you've examined the ovules, you can finish up by dissecting the remaining base parts, like cutting out the receptacle to see exactly where everything was connected.
So, we've taken the flower completely apart. But I want to pause and ask you something.
Why does this all matter, beyond just admiring the flower's beauty or, memorizing a few biological terms? Well, believe it or not, this hands-on process actually gives us the key to understanding how scientists classify the entire plant kingdom. And this right here brilliantly illustrates the scientific significance of what we've just done. By simply counting the floral parts we've laid out on our paper, we can actually classify the plant. If you dissected a lily, you probably noticed the sepals and petals were in multiples of 3. And if you look at a lily leaf, the veins run completely parallel. That makes it a monocot. In contrast, if you dissected a China rose, you'd find floral parts in multiples of 4 or 5, with branched, net-like veins on its leaves, and that makes it a dicot. It's just absolutely incredible how nature organizes itself so mathematically, don't you think?
To wrap up our explainer today, I want to leave you with this really lovely thought from our source material. Flower dissection helps students to develop a feeling of gratitude for the beauty that flowers add to this world. When you truly understand the intricate anatomy, the precise engineering, and the incredible diversity hidden inside just a single blossom, your appreciation for nature deepens immensely. Not all flowers are exactly alike, but every single one is an absolute masterpiece. So, the next time you walk past a blooming garden, ask yourself, what intricate mathematical secrets are hiding just beneath those petals? Thank you so much for joining me today on KognitoKey to uncover these botanical secrets. If you enjoyed this and want to keep learning with me, please hit that subscribe button to join the KognitoKey channel. And please, like and share this video with anyone who loves nature and science. See you next time.